There's been so much attention to CRISPR gene editing now, and of course we're at the conference. How did the Human Genome Project pave the way with its seminal work to CRISPR gene editing?
Right. So the pathway from the pathway from reading genomes, the Human Genome Project, to editing genomes, was, first of all, you need to be able to read in order to edit. I mean, that applies to almost everything. And knowing what is off target requires a knowledge of the entire genome. And so that is how we practice it clinically. Even further back, you could say that the Genome Project led to CRISPR because we were sequencing lots of organisms and we started finding these strange repeats, which looked like junk DNA at first, but in bacterial genomes, but they turned out they were a bacterial immune system, and that led us to do a deep dive and find out that there was something there that could be turned into a technology with some effort.
So can you connect the lines for us from your work to gene editing, which you're participating in now as well?
Right. So when I started my lab, I basically started it on reading and writing DNA and genomes. And that included being able to precisely change DNA, not just make a mess by cutting. Later, it became a little more expedient to do cutting, and cutting involved either meganucleases or zinc finger nucleases or talons or CRISPR, and we worked on all four of them, and were particularly, by the time we got to CRISPR, which was the fourth one, we were fully up to speed, and we were able to, my team was essentially the first to apply them in a variety of systems, including human stem cells.
So CRISPR directly came out of the Human Genome Project.
CRISPR came out of the application of genomics reading technologies to bacterial genomes, and it also came out of work that we were doing on technology development for reading and writing genomes, which was an intimate connection.
One business question now that's a little bit more social than scientific.
Yeah.
Are the Ancestry.com and 23andMe analyses spun off of the Human Genome Project, or is that separate?
Yes, so there are a few direct-to-consumer genomics efforts, including 23andMe and Ancestry.com and Veritas Genetics, and they all capitalized on technologies development, whether it was chips or next-gen sequencing, to deliver high-quality information, sometimes very inexpensively, about things ranging from ancestry to medical information.
Mm-hmm. We're hearing yesterday and today about single strand versus double strand in terms of editing and replacement. What's your thought on all that?
Single strand nicking, you mean?
Yes.
Yeah, so editing gives you the impression, when they use the word genome editing, gives you the impression that you're doing something very precise. But most of the applications, the therapeutic applications, involve fairly blunt and imprecise cut, and then making a mess, knocking out the gene that you're not precisely editing but knocking out. To edit it more precisely, you might use either nicking or a way that you can, without making any cuts at all, make a chemical change, so-called deamination, removing a nitrogen. And the less cutting you do, the more precise it is, but also, so far, the more limited it is. The ultimate is called homologous recombination, where you put in a whole chunk of synthetic DNA. That so far is relatively inefficient, especially in the tissues of your body that are not dividing anymore.
Is that where research is heading, to make that better and more efficient?
I would bet that the main direction that research is heading is to make true editing, precise editing, so easy and so commonplace that we won't be worrying so much about cutting and making knockouts.
Where does gene editing begin and immunotherapy end, or can you, at this point, not really separate the two?
You could still separate immunotherapy from editing a little bit. It is you can make, for example, a very popular one is CAR T, chimeric antigen receptor on T cells, and the key component of that has nothing to do with editing. It's just recombinant DNA, transgenic, where you graft something from a B cell receptor onto a T cell receptor, or otherwise engineer the T cell receptor. That doesn't require CRISPR. But then if you want to make it so it's a universal donor, so any person can donate T cells to any other person, you have to overcome all sorts of other parts of the immune system, and there you might need knockouts. And you can't get knockouts that easily by just putting in extra genes, transgenics. And that's where zinc finger nucleases, talons, and CRISPR nucleases allow you to knock out, or in some cases swap out.